How's it going, y'all? So, welcome to video 2. And as with the first video, I do have a type-up of this video behind the screen. So if it does look kind of scripted, that is because it is, intentionally. I do want to make sure that I don't miss any of the important details when I'm actually going through the topics of the video itself. So for this video, the general outline is going to be a brief reminder of the interrelatedness of cancer and differentiation, as discussed in video 1. Then we'll discuss why it is critical that this is understood by the cancer biology community and implications beyond cancer. I will then outline general mechanisms that I anticipate are broadly utilized during human development. Then we will discuss some of the proteins and protein families that will be mediating differentiation across lineages and where I see important regulatory control schemes. We will then make some connections to central energy metabolism, which will have pretty important implications on the changes in metabolite flow during just differentiation. And finally, we'll discuss some of the open questions I'm working on in better outlining how this system works. So before we begin, I want to acknowledge that I am going to be spending a lot of time discussing cell differentiation as opposed to cancer in these videos. And given that this channel is meant to further your understanding of cancer, this may seem a little bit odd if you haven't watched the first video or read any of the articles I linked in that video. But do remember that cancer cells are essentially just cells that are undergoing a defective differentiation program, and they're cells that are unable to discontinue cycling. This is due to accumulated mutations and dysregulation of the regulatory systems that control the process. And yes, I do know that is a very reductive outline of what cancer is, but it is basically accurate. So the key part to connecting cancer and differentiation is understanding that regulated differentiation, or regulated DNA damage, is essentially a ubiquitous component of the differentiation program. It is core to human development. The mutations and dysregulated DNA damage responses in cancer that drive that genomic instability hallmark are part of this. They are core. The cancer biology community broadly misinterprets DNA damage responses, in my opinion. And as far as I'm concerned, we just need the context-dependent mechanisms by which these pathways are activated to understand the system. So I do want to quickly outline a few reasons this material is going to be important in a broader context beyond just cancer, well, in cancer and beyond, and some of the things that are noteworthy given the content of this particular video. So first, we need to understand how DNA damage is integrated to differentiation in a properly regulated system to understand cancer. This is really important, right? Given that DNA damage agents and DNA damage inducing radiation are frontline cancer treatments. So if we don't understand how the DNA damage coordinates differentiation in specific cell types, there's a chance that these treatments are actually driving the cancer cells towards a more aggressive phenotype by promoting pluripotency and stemness in downstream passages of cell cycle. In addition to the Darwinian evolution we can see going on there based on the selective pressures being applied by these treatments. We do also need to understand how the system works to more effectively target cancer stem cells. Now, beyond this, there are important considerations that do extend beyond just cancer. A big one is going to be in regenerative medicine. So basically, somatic cell reprogramming into induced pluripotent stem cells and the implications this has. So if you're not familiar, these induced pluripotent stem cells have the potential for immune-tolerant treatments through directed differentiation of a person's own reprogrammed cells. We need to understand how DNA damage modifications control differentiation to be able to accurately assess and minimize the potential for these cells to become cancerous with these kind of treatments, and of course to improve the fidelity of the process itself. There should be more efficient ways, I anticipate, than just expressing Yamanaka factors, which are the transcription factors that are used in combination to generate induced pluripotent stem cells. All right, So as I said, understanding the system is quite important, and there is one thing that really needs to be considered as we build out the framework for how all of this is going to work. And this is basically this. Because DNA damage is being utilized to regulate differentiation, there need to be mechanisms by which this can be done without compromising the integrity of the genome, at least in the stem cells. There need to be high-fidelity mechanisms by which the cells can do this, and this is where DNA damage lesion tolerance, DNA damage checkpoint bypass, DNA damage lesion segregation, and multi-passage regulation come into play. At its core, cells seem to mediate differentiation by segregating the damage and sacrificing one of the downstream progeny to protect the genome. And we cannot only consider the consequences of DNA damage in a single passage through cell cycle. Honestly, I do wonder if missegregation of the DNA damage, or maybe just the DNA itself, is central to cancer development. So let's start getting into the DNA damage types and repair pathways that have been shown to be necessary for different types of differentiation. And please do understand that I am still exploring a lot of these networks to further my understanding, so I don't have all of the answers yet, but this is, in my mind, something of a benefit, I think. I like the idea of kind of building out this framework as I explore these networks and giving you some insight into how I broaden my understanding of the system as a whole. What I do really believe is going to be important here is looking at repair pathway choice and nuclease recruitment to chromatin in a cell cycle phase in a passage-specific manner. This is going to help us understand lineage-specific requirements in these cells. Cool. So here's how it seems to work. Basically, specific DNA base modifications and possibly often the generation of A-basic sites are what are going to be central to guiding differentiation. I mean, yes, this has been established in a few systems, right? But it will probably be much closer to a ubiquitous occurrence than what is currently appreciated. As an established example of this, activation-induced cytidine deaminase, AID, mediating somatic hypermutation and class switch recombination. This is observed. And as AID is a member of the apobec family, this is going to require a focus on other apobec family members. As I think of it, if it happens twice, it happens 100 times in the cell, just in slightly different ways. Not literally, of course, but it's a fairly good guiding principle into understanding the redundancy of life. So let's talk about the specific base modifications. Cytosine, as well as guanine modifications, are going to be central to this process. On the cytosine oxidative side, which is where my current focus is, we should be focusing on the progressive oxidation of 5-methylcytosine. This occurs downstream of DNA methyltransferase activities that methylate cytosine, and is going to be mediated by our 10-11 translocases that I mentioned in the short, TET proteins. And we would expect thymine DNA glycosylase to be a central facilitator of the repair of these enzymes, as that has been an established function for this protein. On the guanine side, some of the regulation of differentiation seems to come from 8-oxyguanine modifications, and mitochondrial ones at that seem to be playing an important role. I need to look more into this, honestly, and it'll probably be covered in a future video, but it would be interesting to see if accumulation of these mitochondrial 8-oxyguanine mutations regulate mitochondrial maturation and segregation during mitosis. It's not random. I expect that base excision repair machinery is going to be profoundly important. Generally, and possibly more so in less differentiated cells. However, transcription-coupled nucleotide excision repair likely plays an important role too, and understanding the interplay, especially given the degree of differentiation as they progress through the system, is going to be very, very important. So let's talk a little bit more about the TET enzymes, of which there are three family members, TET 1, 2, and 3. These proteins are dioxygenases that progressively modify 5-methylcytosine to 5-hydroxymethylcytosine, 5-formyl-methylcytosine, and 5-carboxylcytosine. During development, as expected, we see dynamic modification to the DNA that are driven by these enzymes. For example, global accumulation of the 5-hydroxymethylcytosine during neuronal differentiation in the mouse brain has been detected. TET2 and 3. TET2 and 3 inhibition leads to neuronal defects. TET2 knockout disrupts megakaryocyte and erythroid differentiation, and TET2 regulates myogenin transcription in differentiating myoblasts. And yes, the 5-hydroxymethylcytosine mark has been shown to be asymmetrically inherited in dividing stem cells, with elevated 5-hydroxymethylcytosine being associated with the immortal strand. Though that hypothesis has been challenged, and I have not spent enough time looking into it to speak on it really at much length at this point. It is important that TET activity seems to be skewed towards S phase, which is somewhat aligned. Because early embryonic stem cells do not have gap phases, we would expect to see much of the basis of the regulation of differentiation being coordinated in either G2M, or rather coordinated in S phase during the synthesis of the DNA, G2M at that barrier, or if we're talking about early embryonic stem cells, possibly the S and M barrier, and whatever responses could be regulated at that point. So, speaking of S phase, I think we as in like the scientific community, very much misunderstand what is going on during replication fork stalling and collapse, and the regulatory control during these events. I will probably be looking into this in the future as the significance becomes more apparent, but here's the thing. If during differentiation we need to mediate altered epigenetic changes to one complement of the DNA, where better to do this than during the synthesis of the DNA during S phase? Of course, mitotic DNA synthesis is a thing, and the implications could be very interesting, and it'd be kind of cool to see if they're associated with caspase-2 activity, but that will be probably discussed in a future video. So, on the metabolic component of using DNA damage and base modifications and the proteins that are going to be involved in this to coordinate the process. We know that obviously during cancer we've got the Warburg effect, we've got that glutamine dependence, but stem cells do have altered metabolism too, and some of the subtleties and the changes could be a little bit more nuanced than what we're currently appreciating. So I want to kind of discuss how some of these proteins that are going to be involved in the DNA damage and the epigenetic changes do intersect with cell metabolism. So TET proteins and Jumanji demethylases are very interesting because they use alpha-ketoglutarate for their reactions, which you will remember is a critical intermediate in the TCA cycle, and through a couple conversions downstream of glutamine, you can get anaplerotic entry of this metabolite into the TCA cycle for a variety of purposes. So TETs and Jumanji demethylases use alpha-ketoglutarate. And of course that glutamine dependence is going to be interesting. And of course, isocitrate dehydrogenase 2, I believe, is mutated in some cancers, neuroectodermal ones, I believe, but I'm not sure how broad that mutation is. These do alter TET protein functionality, and I believe the different metabolite that is generated because of the mutation in isocitrate dehydrogenase 2 does change the regulation of TETs and inhibit at least one of these proteins. Okay, now on the histone acetyltransferase activity, like these proteins use acetyl-CoA, well understood to be involved in the TCA cycle. PARP1, which is involved in single-strand break repair and chromatin remodeling, consumes NAD, the oxidized version, as do sirtuin deacetylases. Even pyruvate kinase can function as a protein kinase in the nucleus to regulate transcription. So when we are considering differentiation as a whole and the chromatin, transcriptional, and metabolic shifts that occur during this process, it is going to be important to appreciate that at key points, the regulation of metabolism and metabolite flow may look very different from normal. And with the metabolic shift you will typically see to oxidative phosphorylation during terminal differentiation, it's not surprising that this could be coordinated along with the transcriptional changes observed. I expect that mitochondrial fission-fusion dynamics, mitochondrial ER-associated membranes, and, as I mentioned previously, selective substrate release from the inner membrane space are going to be very important here. All right. So where my current focus is, and the things I'm kind of looking at and will be outlining in future videos, depending on which questions seem to pan out, are as follows. Basically, and do bear in mind that there's a good potential that these questions have been answered, or the mechanisms that would give us the answers have already been outlined. I certainly hope so. I just may not have come across the answers, so do not assume that these are unanswered questions, just that I do not know the answers myself. So the first one is extremely important, or intuitively I expect it to be. And that is like, why do some transcription-coupled nucleotide excision repair defects not lead to elevated cancer risk? What are the implications of this? And how do bulky versus non-bulky lesions differentially regulate the system? That interplay between the BER and the NER repair machineries seems like it's going to be very important. I'm not sure about the global NER, but transcription coupled NER seems like it will be very important. Okay, so 2, can cells intentionally mutate the DNA sequence and then asymmetrically segregate the damage such that a regulatory control scheme would be restricted in the offspring? How might this concept of asymmetric lesion segregation intersect with homology-directed repair? And what does this imply about the control of end resection and the interplay between 53 binding protein 1 and BRCA1? Stem cells have been shown to use a higher fidelity form of non-homologous end joining. How does the end... So there's a lot going on here. I am not sure how well the interplay is going to pan out, but it does seem very important in looking into this regulation, understanding cross-passage control. All right, so 3. What are the consequences of caspase-induced DNase mediating single-strand breaks at the G2M checkpoint in cell cycle passages after a cell has been subject to DNA damage in the preceding passage? How can G2 regulation of the following G1 come into play? Which could be basically RAS induction in G2 can lead to the upregulation of cyclin D1, which is canonically associated with that early G1 phase passage through cell cycle. But this leads to having cells that are prepared to continue cycling in the following passage, where you would have lower levels of the cyclin dependence kinase inhibitor P21 and higher levels of cyclin dependent 2 activity. And of course, cyclin dependent 2 is in complex with cyclin E, mediating progression through late stages of G1. Now, we see that cells who do inherit that 53 binding protein foci resolve that damage prior to S phase commitment, and given that TETs have been shown to have to function largely during S phase, at least in some of their canonical functions, that seems like it's going to be very important. So for how might the use of DNA damage, which is often tied to transcription of lineage-specific genes, promote the specific mutations that are associated with cancer? There's a lot to look into this, but when we're looking at these like glycine to valine mutations in like exon 2 of RAS, and we see these very specific mutations in different types of cancer. The explanation that's kind of been outlined is that these mutations provide a advantage to the cells, often through the driving a hyper-proliferative phenotype, but I'm not very convinced in this, and I don't have the statistics background to really be able to validate this, but if you're looking at a system where things are done at random, and just the scope and the scale of the genome, random doesn't seem to make sense. If this system is controlled through DNA damage, something about the regulation might be selecting for or potentiating these specific mutations. I don't know what that regulation is, but it could be present. Cool. Now, I know that TET proteins, DNA methyltransferases, neoproteins, which we haven't really covered much, apobex, DNA glycosylases, they may not be overly familiar to you at a detailed level, and you may not have even heard of a lot of them, which is kind of the nature of how we study the cell, given how specialized everyone seems to be. I can say they're not as familiar to me as I would like them to be, and I've only really recently come across them this year. But there's not that many, so it really shouldn't be that hard to put together. I always kind of think of these when we're looking at maybe a couple dozen proteins and what they're doing. I always kind of think back of like when I was taking courses in school, we would learn the entire process of glycolysis and regulation in a single lecture. 10 enzymes plus all these different metabolites, and that would only be a small component of what we'd be tested on. So if we're looking at 3, 4, 5 protein families, a couple of dozen interactions, that doesn't seem outlandishly difficult to outline if we have an understanding of what the underlying regulatory scheme that is governing differentiation is. We just have to slot things into the appropriate space and figure out what the implications are to their activity. But what seems to be core to this is going to come down to repair pathway choice. And that should be guiding our understanding of what types of cells during which passages of differentiation use which repair pathways. It should be very insightful. So, this is really just the first video as I'm going to look into this a little bit more, but generally this is how it's going to work, right? To my understanding. DNA damage, base modifications, potentially the generation of abasic sites. I expect lesion segregation is going to be very important, and homology-directed repair could be as well, though the type is going to be very interesting, and how this ties into things like alt mechanisms of lengthening telomeres versus the use of telomerase, or the induction of telomerase, and how specific types of cancer and or pathways of lineage specification intersect are going to be very interesting. So those are the kind of things I'm going to be looking at in the future videos and outlining for you. So very brief, not too much going on here in terms of like the specifics yet. We're going to get there. But if you have any questions, I can elaborate on basically any of this and link it to the systems we described in the previous video about the intersection between death and differentiation of the proteins that govern each of these interrelated processes. And of course, I do want to say, if anyone has any kind of inkling as to what protein mediates cleavage of BID to JBID, let me know. When I'm looking at this system and also considering the death, differentiation, inflammation dynamic, JBID is going to be very important, I anticipate. So if anyone has any insight, please let me know there. Any questions about this overall system, let me know. But this is going to be the general framework. Thank you.